So welcome, nice morning here, and we're going to have a good morning with Graham speaking today. I'm really quite flattered and feel honored that Graham asked me to introduce him to you today, and even going to the extent of changing the date of this conversation. So I really appreciate that. I've known Graham for about 40 years, since he arrived here at MIT. And he and I have a few things in common. We both have a kind of a chemistry background. And Graham did his PhD with two people I got to know quite well, Olke Uhlenbeck and Nelson Leonard at the University of Illinois in Urbana. And after that, Graham went to Bruce Ames's lab for his postdoctoral work, and then he came here. And that was in 1975, 76.
I'm just going to see if
All
I
right,
can figure out what just happened here.
Yeah, okay. At MIT, Graham has proved to be a stellar scientist, working on two different areas at the same time, DNA repair and mutagenesis on one hand, and symbiosis between bacteria and plants on the other. And he has done phenomenally well in both areas. He's also trained many students and postdocs who've gone on all over the world and established great programs of their own. So that's a real credit to Graham. Now, I'll just give you list a few of his accomplishments early on, not the whole list because it's a long list, but I'll just give you two. And they include DNA damage-induced changes in gene expression, and the work that he did in the lab that led to the discovery of translucent DNA polymerases. And you folks in the field know that these are really, really two important things that he did. On the symbiosis front, his work has led to the discovery of a new step in vitamin B12 biosynthesis. That was the missing step in vitamin B12 biosynthesis. And of the discovery of a ribonuclease that plays a critical role in RNA metabolism, ribosomal RNA metabolism in bacteria. So these are another new things, and the list goes on. And in recognition of all this, Graham recently received the prestigious NIH Outstanding Investigator Award. And this is a really great award. It lasts for 8 years and comes with good money. And so I think it's quite, and I think it's quite likely that Graham is going to go on for quite some time. Now, we all know that Graham is also devoted to teaching. He has taught 721, of course, for many, many years. And he's taught 7014 for as long as I can remember. And 7014 is this introductory biology course that many of our students take. And I personally found that teaching 701 series of courses is probably one of the more difficult of the things that I've had to do. And so I was quite pleased when I got out of it after a few years. But Graham continues to soldier on and never complaining about it and still doing a great job. He also ran the undergraduate biology office for many, many years. And he's also a director of a HHMI-funded educational group that he's going to talk about today. So he has done a lot in the area of teaching also. And for that, his institute has recognized it through a Margaret McVicker Faculty Fellowship that he had for 10 years. Now, outside MIT, Graham is also very actively involved in ASM, American Society of Microbiology Affairs. He co-authored the book, the famous textbook, DNA Repair and Mutagenesis, that a lot of people have. It's a must. And then he also was the chief editor of Journal of Bacteriology for many, many years. So that's a lot of work that he does. So the question really is, how does Graham do all this? I've not even told you about the fact that he's interested also in music, and he takes music lessons every year and things like that. So for the last several years, I've had the pleasure and privilege of collaborating with his lab. So I got to know him a little better, got to know how he operates and things like that. And what I've found is that the reason why Graham can do all of these things is that he has this knack of doing multiple things simultaneously, all without losing his cool, always with a smile, and making it all look completely effortless. So that is the real Graham, and he's a real remarkable person. Now, Graham is also someone I would say is a real gentleman, all right? Okay, so I have played squash with Graham for many years, right? And squash is a game. Some of you, I'm sure, know it. That it's a game played, with court with 4 walls, and the game can be fast and furious, and it can be quite intensive and quite challenging if it's very, very competitive game. And the goal is to hit the front wall before the ball bounces, and then see what happens, right? And the other thing is that if you get hit while the ball is going towards the front wall, the point belongs to your opponent. So there's something to do for hitting your opponent, right? And I have to tell you that in all the years I played with Graham, he has never hit me once. Yeah. And I have come off this, the squash courts with welts on my back, even the back of my head, but not because I was playing with Graham. I had played with somebody else. And so, Graham, I think, is one of these people who believes in really, it's really less important to gain a point than to take the risk of hurting your opponent. And as the British say, Graham, that is a real sportsmanship and being a real gentleman. Yeah.
Thank you.
So, on a personal note, I would like to end on is Graham also has been an extremely kind, generous, and a true friend to me over the years. A recent example of this I'll give you is that I closed my lab just a few months ago after 60+ years of doing research. And I think some of you might know or will come to know that it's a difficult thing to do. But Graham was very sensitive to that. Even before I closed my lab, he invited me to his lab gatherings, the lab celebrations, and that was very kind. I would get notes from his administrative assistant, Mary, saying that, Wednesday at 2 p.m. is pie time. Please come, and things like that. And I've taken advantage of that act of kindness and enjoyed being with the Walker group. And so, I think, thank you, Graham, for this very, being a friend, for this act of kindness and being a friend all these years. And so the floor is yours, and we'll look forward to hearing from you.
Thank you. Let's see, turn this off, maybe. Well, thanks, Tom. Apart from everything else, when I first set up my lab and I was standing here in this empty lab, the very first person who walked in was Tom to say, so we go back a long way. This Conversations with a Scientist, Munda and I conspired to start this series. I'd always wanted to do it, and one year we had some money left over from this large Howard Hughes HHMI grant for education that I directed for 25 years, but at the time it never occurred to me that I would actually have to talk in the series, so I guess it's finally happened. Anyway, what I'll try to do today is just sort of give you a sort of broad sense of my life, how I sort of got here, and how I've seen MIT, a little bit about the research while I'm on there. Tom alluded to the fact that there was this dual sort of interest in the lab, which to me never seemed all that different apart, and realize it looks a little farther apart to the rest of the world. And just try and give you some pictures. I found a bunch of pictures, so I have some slides, and I'm just going to mostly try and tell you stories, some of my favorite stories as I go along. But I was born actually in Arlington, Massachusetts, but of Canadian parents who were just here very, very briefly, and they went back when I was 3 months old. So I grew up in Ottawa, Canada, and I had two younger brothers, one 2 years younger, one 5 years younger, and a sister who arrived. She was 11 years younger than me. So there's some early pictures of the family when I was growing up. I had a nice life. I was in Cub Scouts and later Sea Scouts. I played church league hockey, but not very well by Canadian standards. I learned to play violin in the Ottawa public school system, which was my entree to music. I thought there were two sort of influences that were important, headed me towards my MIT career. One was the one point, the family bought a piece of land on the Gatineau River north of Ottawa. It was about 20 miles north of Ottawa, and we had to row about a half mile on a wooden rowboat to get to the other side. And my dad, who was not, he was a statistician for the government, and my mom actually was trained as a biochemist originally. My dad figured out how to build a kind of rustic cabin with just him and the 3 little boys using counterweights and stuff. So I spent my summers, this was just a few feet from the edge of the water, in the Canadian woods, wandering around, and a lot of swimming and fishing, but also looking a lot at nature. I think that's, where my interest in biology originated. I also, the other major influence, I think, while I was growing up was that I had a chemistry stat, and in all honesty, I mostly liked to burn things with colored flames, and I did that a lot. And after a while, I got a book from the library. I learned you could decompose hydrogen peroxide and get oxygen. Then you could burn things in free oxygen. And I was consumed with wondering what it would be like to breathe free, just oxygen. So I saw that if you had a hydrochloric acid or dry cells, you could decompose water into hydrogen and oxygen. I had neither of those, but I thought salt might work, and I had a toy train transformer. So I hooked that up and let it run and collected some gas and picked up the one that I thought probably was the oxygen in it and took a deep breath, only to discover I had produced chlorine. It was one of the great epiphanies of my life as I coughed around the cellar for 15 minutes wondering I was going to die. So that was the beginning of my sort of interest in chemistry, I guess. Then I went to a local high school, Glebe Collegiate, and I was a gymnast. We had a pretty good team. We won the provincial championships one year. They didn't have strings, so I had to learn to play brass instruments. And I learned euphonium, and that was actually a big event in my life because after I'd been playing a couple of years, they had a mass band concert where few selected players from each high school, and the person I sat beside was Jan, who later became my wife. And it was not love at first sight on either side at that time, but I did notice that her euphonium played about a fourth higher than mine did at the start of the day, and I realized there were some higher notes than I realized. But actually, Jan used to, the big music festival in Ottawa, and Jan used to win the music festival. Her picture would appear in the paper, so we sort of remained rather dimly aware of each other to the beginning of university when we kind of remat and started dating. So music was a big part of my life. And at that time, too, I also picked up a guitar, which everybody was doing at that point. And that actually is an instrument that stayed with me through the rest of my life because it's such a social instrument. I had... When you went to high school in Ottawa, there weren't the number of electives you guys are used to. There were 2. If you wanted to go to college, you took Latin, and if you didn't want to go to college, you took shop. So I took Latin, and I actually enjoyed it, was pretty good at it. So when I went to university, the big decision was, should I go into science or should I go into classics? And I think I made the right decision, reasoning that I wanted to go into a field where it was all going to happen while I was there instead of a field where everything had already happened beforehand. And my mom got a rather debilitating illness during my senior year of high school, so I decided to stay in Ottawa and live at home because of that. And I went to Carleton University in Ottawa, and my thought going into it was that I would do quantum mechanics. I didn't quite understand what it was, but it had a nice ring to it, and I knew it was sort of chemistry and math, and I thought I was fairly good at both. But then an unexpected thing happened. I had to take an introductory biology course, and I ran into DNA, which was not in the high school curriculum at that point. And I thought, that's the coolest molecule in the world. I want to work on that. And I The only thing I understood about quantum mechanics at that point was that DNA was too big for quantum mechanics in the mid-60s. Computing wasn't just up for it at that point. So I said, I'll become an organic chemist and I'll synthesize it. And that was sort of how I ended up going into chemistry and into particular into organic chemistry. And that's where I encountered the first of my really important mentors, Bob Whiteman, who was in the department. He let me do a little tiny project with a nucleotide in a third-year lab, and then let me do an honors sort of project with him, sort of senior UROP equivalent with him, and which I was so excited. I grandly entitled it Studies Towards the Synthesis of Ribonucleic Acids. One of the things I did was to make a methylated, 2' methylated ribonucleoside. There are a couple of things sort of looking back in retrospect that are interesting about some of that work I did. One was I started out by synthesizing several hundred grams of nitrosylmethylurea, which is an extraordinarily powerful carcinogen, but it was made at the time, used at the time in labs because it crystallized really nicely. I was dimly aware of that, so I think I wore gloves or something. And then the way you methylated it was you had an ice bucket on a stir, and in that was a solution of base and ether. And with a safety shield, you added little bits of this stuff. You were generating diazomethane. And as you can, down at the bottom, there was a little note. The diazomethane is very explosive, so the safety shield, glasses, and gloves should be used. The whole thing blew. I was lucky that I got a little singed, but nothing more. There was a big investigation, and they decided it was the thought was that the magnetic stir had sparked the explosion. So the suggestion was the next time I not use the magnetic stir. Instead, I do it in an Erlenmeyer and shake it like this. And I think I'm still here today thanks to Bob Whiteman volunteering to step in and tell me he would do the last reaction I needed to get the last bit of material instead of me. So I was very, very grateful for him for doing that. Bob was also very important to me because he, well, two things. One, he arranged for me to work at National Research Council in the lab of Saran Narang, who Tom knows, who was another of the tiny number of groups in the world that were sort of vaguely competing to synthesize the first gene. And I was so excited that I was in a lab that was trying to synthesize DNA. The other thing Bob Whiteman suggested, I was trying to think about where to go to grad school, and he thought that Nelson Leonard at the University of Illinois, who Tom mentioned, would be a good choice. Nelson was a very famous organic chemist. He'd synthesized chloroquine, for example, the anti-malarial, and he was starting to work on nucleotide chemistry, particularly cytokinins. And so I thought that, in the end, I went to University of Illinois specifically to work with Nelson. Nelson was remarkable, though, and he had another influence in my life. He was a singer of quite some talent. He sang solo at Chicago Symphony, and he was actually offered 2 positions at Illinois, one in one in chemistry and one in music. And that was when he finally had to decide which career direction to go. And if any was gentleman, Tom thinks I'm a gentleman. I'm a pale imitation of the graciousness that Nelson had. I didn't know this. I think he really influenced me with his singing besides the science, though, because I later went on, started writing songs in grad school, and later on going to music camps and performing and some stuff, and then getting some voice lessons. And now Jan and I sing with a sea shanty group based up in Gloucester called Three Sheets to the Wind. We perform maybe 20 times a year at tall ships festivals. We're singing at Rockport First Night. We sing at the Crane Estate up in Ipswich last week. It's been a lot of fun, but I trace it back to, I think, to somewhat the influence of my PhD mentor, besides his influence on science. Then the other thing that happened was, well, during my first year, Aki Uhlenbach, who Tom mentioned, interviewed on Job Candidate. And I was captivated by him when he was hired. I immediately bustled down to see if we could set up a little project on the side, which was fine with Nelson. And we started on some little idea I'd had, but then one day both of them walked into the lab waving the latest issue of Nature, which had a scheme for being able to expand the number of ribonucleotides you could make, which at that time, the state of the art was to run polynucleotide phosphorylase backwards and then use various enzymatic tricks to make oligos. And it was simply, the idea was to put a 3' block so that you could, 2, 3' side of the ribo to block it, and then remove the blocks. You could sort of add things one at a time. And so I went roaring down to the stock room late in the afternoon, synthesized the thing, and arrived the next morning to give it to Aki to try the reaction. I didn't realize at the time that wasn't usual behavior, but anyway, it was a good start to our thing. And so my thesis was partly thinking this. Did some work on cytokinins that sort of demolished their theory that they thought that they work by being minor bases in tRNA. I worked on some cyclic nucleotide analogs. I did this big project with the making RNAs, and so I had trouble coming up with a title. It had to be pretty general. But there's one other thing in my thesis, which I've probably shown everybody that worked. Nelson consulted for Lilly, and at one point they got interested in three alkyl adenines because they thought they might be antivirals. Well, it turned out these weren't too hard to make, so I went down to the old chemical room and I got all of the alkylating agents I could and moved them up to my lab because nobody was using them. And whenever I got depressed about how science was going, I'd synthesize a few of these. And so if you look at my thesis, there are a whole pile of these. They wouldn't even all fit on the page, but it sort of told me that, there are setbacks and things, and you have to just sort of keep going. I had a picture of back from Aki's group there. I didn't have any actual pictures that I could find easily from Nelson's group, but one thing I did do at some point, I took over putting up the announcements for group meetings. So I have these images from some of the fun we had thinking about science back then. There's one time when Aki, who was very tall, and I, who was not tall, both spoke together one night at the Nelson Leonard group meeting. It also reminds me that because it was a chemistry lab, there was a lot of running water and hoses everywhere, and so floods were a fact of our existence. And in these little cartoons I would do for our group meetings, they became Urbana by the Sea. And so we had, as I was thinking about this talk, I found this file of research group meetings that I had and remembered all the floods that we had. I think you could tell I had a pretty good time in grad school, and I discovered we had to do these quarterly reports, successes, failures every quarter, and give them to them. And I found even those could be fun because one time we'd had a group picnic, and I'd made something called butter tart squares that I knew Nelson Leonard had particularly liked. So I wrote up the synthesis of butter tart squares and put them in the quarterly report and didn't say anything. And about 6 months later, I came in and there was a note at my desk from Nelson, who was Tom, very gracious man. He said, "Please submit samples for testing prior to publication." Apparently someone had called in, and he hadn't looked at my report, of course, and somebody called up asking how to make something. He'd fished it out, and while on the phone, had discovered this. So for the rest of my time there, whenever I handed him something, his job was to find what I'd hidden. And when I handed him my thesis, he was in my office about a minute and a half later with the offending page. I was felt very lucky, though. Jan and I got married just before we went to grad school. We lived in an apartment immediately under the university power plant, but we rented land from the park district, and we had a wonderful garden. We had a big influence on me there was music. We didn't have all that much money, and there was a wonderful folk music program on the student radio station done by a guy called Rich Warren, who was trying to sort of emulate a program that folk music been going in Chicago and WFMT for many years called Midnight Special. And by going down to the radio station, we ended up being friends. He's been coming to Thanksgiving. We frame shift Thanksgiving to Saturday, so friends can come after their own Thanksgiving. Rich has been coming for more than 30 years to share Thanksgiving. But he did a lot of series of concerts, and so all of that was very inspirational for me. I heard a lot of music. I started writing my own songs. We used to take my guitar to departmental parties, and it was a less politically correct world. I had a book of slightly off-colored songs that my brother had spotted in the library when he was down, so it was in my guitar case, and then the chair of the biochemistry department got hold of it and started bellowing these things out. So my job at the departmental parties was to play guitar while we sang all sorts of songs. Anyway, life was full of music and friends. And I think what I learned out of this was that the groups I were in were like a family. And I thought it was, even though there's setbacks, there's a way to do science where it's a lot of fun and everybody supports everyone. And also, I think it's important to have balance in your life. And so I've sort of tried to follow those lessons ever since and to pass them on to the people that I've had. So by that point, I was thinking that I didn't want to be one of these chemists that only did chemistry and had to ask a biologist to explain it to them. So I decided I needed to learn genetics, and I thought about several labs. The one I eventually chose was Bruce Ames at Berkeley. And I think, in truth, the reason I decided to work with Bruce, I'd heard him give a talk when I was a grad student, and while he was down there talking, I thought, that man thinks like a cell. I want to learn how to think like a cell. You could almost see him as if he was inside the bacterium thinking about how things worked. So I went to Bruce's lab just at the time that he'd done the work. He'd done a lot of work in histidine biosynthesis, but as many of you know, he developed the very first short-term test for potential carcinogens and mutagens. And it hadn't been working terribly well, but then he found that if he put a naturally occurring plasmid in it, suddenly it worked a whole lot better. And if the strain had the plasmid, it was a whole lot more mutable with lots of different things. And so I thought, must be something interesting on that plasmid. I don't know what it is, but it's something to do that damaged DNA is getting changed into mutated DNA, and we'll find out what it was. So I started to work on that, and that pulled me into the discovery that this was somehow linked to what was beginning to be called the SOS response, some kind of induced set of diverse metabolic responses. Turned out induction was caused by DNA damage. And I had fun with Bruce as well. I learned through a technician that it was possible to, if you got up really early in the morning, you could take a boat out from Emeryville and go salmon fishing outside of the Golden Gate and still be back in the lab before most of the postdocs had arrived. So occasionally I got salmon fishing as well, and continued to play my guitar at departmental parties. And I seem to have lost it, but I got the 4-letter word Poet Laureate Award for one night for repeating a few of the songs that we used to sing in Illinois. But anyway, the joy of doing science continued. I was also very lucky that I sort of acquired two more, I think of them as mentors. One was Evelyn Witkin, who some of you may know got the Lasker Award along with my former grad student Steve Allage a couple of years ago. Evelyn had somehow, through the grapevine, heard about my work and showed up one day in my lab while I was a postdoc and wanted to talk to me. And later, when I came to MIT, I found I needed to get something called a Latarjet meter to measure UV dose, and I couldn't figure out where to buy one. So I called Evelyn, and she said, "Oh, that's because Latarjet makes them in his basement in Paris. I'm going over there, and I'll let him know you need one. I'm going over in a month. I'll bring it back for you." And she brought it to my office. It was extraordinary. Phil Hanawalt was the other one. I went to my first DNA repair meeting, and I met his grad student, Priscilla Cooper, who became a lifelong friend, who took me over and introduced me to Phil, who sized up immediately. I was on my own. I was coming from a lab that didn't need DNA repair and kind of made me a part of his group, welcomed me in. And that was just from his, he's 80, he's retiring. I think it's 85 or something now. Except if he gets his grant renewed, he's not really retiring. So anyway, that was, he inspired me in part because both he and Evelyn believed very much in a community that shared information and where everybody talked and shared ideas. And Phil did that, but he also organized some very large meetings. And eventually, as I'll tell you later on, I organized a few meetings that sort of followed through a thread of meetings that continued a thread of meetings that Phil had done. So things were different then. I was a year and a half there, and I ended up interviewing for jobs. I didn't have any papers then. It was different. MIT didn't advertise at that time, so we only heard that there were jobs at MIT when I think Malcolm Gafter came and mentioned that when we were out there. So several of us came at the end of the job search, and in truth, I loved this place. I would have worked in the bathroom, which was probably a good thing, because when I went to talk to Boris Magasanik and Gene Brown, who were the chair, and I'm not good at negotiating. I always get my guys to talk to somebody else. So I sort of diffidently asked, just curious what they did in the way of setup funds, and Boris looked at me and said, "At MIT, we don't have setup funds." And I said, "Oh, sorry, I didn't mean to mention it." And it's okay, you don't work in the bathroom, you know. Anyway, so I loved the mix of science teaching, what I saw here. David Botstein dragged me off to the project lab while I was here, and I carried incubators around. I was completely sucked into the idea of teaching in the MIT style. I even got lost trying to come back from building 4 and wondered if that was part of the job test, was whether you could find yourself back to headquarters. Anyway, I left here and didn't know what the odds were, but there is a good story about when I got offered the job where everybody in Bruce's lab knew that I had really loved MIT, and there was a little lunchroom surrounded by the two lab rooms, and the phone rang, and somebody said, "Graham, it's MIT calling." And so I went, and I listened, and everybody lined up at the 2 doorways. And Boris said, "Graham, it gives me great pleasure to offer you a job." And I went, "Yippee!" And everybody came piling in. I scored a touchdown, and about 20 people landed on top of me. The phone got lost. It was under the table. Eventually, we unpile, I get up, and I pick up the phone, and I say, "Hi, Boris, sorry about that." Boris says, "I take it you're interested." Said, "Yes, thank you so much, goodbye." And then for the next week, it was really interesting because all the senior Berkeley faculty who had not talked to me very much came along, and every single one of them came and pushed me into a corner and said, "I'm going to give you the most important piece of advice you ever got in your life. Get it in writing." So this went on for a week. So finally, I called Boris back, and I said, "Yeah, I feel really silly about this, a little awkward, you know, but I'm getting all this advice from the senior faculty. Get it in writing." And Boris said, "That would be just fine, Graham." He said, "What exactly would you like in writing?" I said, "Oh, God, if I can't trust you guys to take care of me, I don't want to come." So anyway, I just came here, and I feel like a lucky star hit me on the head, and I've been here ever since. And one of the great joys to me, Bob was hired in that same job search, and his lab was beside mine, and he arrived just a few months after I did, and we shared. And I can't name everybody who's influenced me, so I apologize to whoever it is I'm forgetting, but I just have to mention a few people that really influenced me over my early years here. Boris and Jean, I've mentioned. Maury Fox was a huge influence on me. Mary Lou Pardue, Salvador Luria, Tom, as I've mentioned, Lisa Steiner, David Botstein, as I've said, Vernon, who was just down the hall from me, Phil Sharp, Tony Sinsky, who I got to meet by just the fact that I passed by his office. Barbara Meyer shared an office with me, Monty Krieger, Lenny, who's here. I was in between Bob's lab and Paul Schimmel's lab, so I got to know Paul. And in a funny twist of fate, I got to meet two people who would become faculty. I met Leona Sampson at a Cold Spring Harbor meeting. A couple of weeks after I got to MIT, I went down and I met her. She was working with John Cairns. Later, she came here, and I met Sue Lindquist very, very early on at so-called heat shock meetings. And as those of you who heard my tribute to Sue, we were sort of dancing buddies at the heat shock meetings. And there were so many more people have influenced me here. I did get eventually a little group is going, and I found Pam Langer, transferred from another lab. Bill Shannonbrook was my first grad student out of the pool. Cynthia Kenyon transferred from another lab, and then one year I had Steve Elledge, Steve Winans, and Karen Perry came in, and we always had a number of undergrads, and there's Steve Elledge tossing a piece of pizza dough at one of our early group parties. I had an amazing, after a small number of years, I had amazing administrative assistant Marianne White, who finally retired in 2006, who many of the people in the department went. She was a big mother to all of us and a source of warmth and joy. And Mary Gallagher, I think, has stepped into that role very recently in our lab, and we have another good era of that lab. But anyway, we started to just work on this PKM 101 stuff, and it was clear that whatever it was doing, but mutagenesis was somehow induced by DNA damage. And around that time, Malcolm Casadabin and Stan Cohen made a derivative of phage mu that had a promoterless lac. And mu can go in randomly, so if you let it insert in the chromosome, you could make fusions to essentially anything. And it was Cynthia's idea. She heard about it at a meeting to try and just make some random fusions, and we see if DNA genes were turned on by DNA damage, which was a rather controversial idea related to this SOS response. Seemed really straightforward to me. I was only worried about whether the genes were there, and as it turned out, there were genes induced by DNA damage. And I guess looking back at it, I think this was the first screen that looked for genes that were searching for genes on the basis of their regulation instead of the basis of their phenotype. And of course, something we now do all the time with first microarrays and now RNA-seq and stuff like that. But it was new at the time, and I thought it wasn't all that exciting, so I was going to publish it in Journal of Bacteriology, but thankfully Cynthia persuaded me it was more important and not as obvious as I thought to the rest of the world, and so we published it in PNAS, and I think it's probably the paper, certainly one of the papers in my career has got most of the attention. The one thing I did do was try to get rid of all the bacterial jargon, use regulatory network instead of Regulon, and I think that maybe has helped with it as we went along. And the other sort of theme that, we followed was trying to figure out what the genes on the plasmid were, and we found that actually they were able to suppress the deficiency of genes in the coli chromosome that if you lost them, you became non-mutable. And Steve Elledge did a rather heroic job of cloning these things, which was tough both because of the restrictions on recombinant DNA in Cambridge, and eventually did positional cloning to find the genes on the chromosome, which turned out to correspond to the same genes that were on the plasmid. We didn't know what they were, but we knew their function was to take damaged DNA and make it into mutated DNA. And Ben Lewin, who was at that point the editor of Cell, had asked me why I hadn't published our Cynthia's paper in Cell. well, they didn't seem to be publishing hardly any E. coli papers. So anyway, when Steve finally cloned this gene, I tried there, and he got right back to me and said if we could just put a single paragraph in about what its function was, then it would be acceptable. Well, it took 17 more years before the field figured out what the function was. But it turned out at that point that UMUC and all these other things that were showing up everywhere were all, as Tom mentioned, the translesion DNA polymerases of the Y family, and UMUC was required for mutagenesis in E. coli. The other partner in there, UMED, we actually were able to work on it earlier because it looked like the back end of the Lexa repressor and lambda repressor. And those of you may know both of those repressors have a potential autoprotease activity that gets activated when they bind a single-stranded DNA that's coded with REC-A. So it turned out that the SOS response was important not only to turn on genes at the transcriptional level, but that overlaid on that was a post-translational form of regulation that went on involving this cleavage. And actually, I knew where Mundana's worked years before she came here because she worked on stuff very closely related to this. We've continued to sort of, that theme, there was an awful lot of stuff. I don't have time to go through it all, but just give you a couple of sense of where that work has led to, that it still showed up. It turned out that UMEC is a bacterial invention, but there's a member of this family called Rev1, which is required for mutagenesis in eukaryotes, takes damaged DNA, makes it into mutated DNA. There's also Rev1 and Rev3 and Rev7. Rev3 is a member of the B family. Rev7 is a hormodomain protein that's a sort of polymerase accessory protein in part. And you need these three functions in eukaryotes in order to take damaged DNA into mutated DNA. And there's something I'd thought about for years and years while we were working on this phenotype of non-mutability in bacteria, but I'd always wondered about this sort of, whether it was potentially germane to this situation, which all too familiar to you, cancer, give somebody chemotherapy with a DNA damaging chemotherapeutic agent like cisplatin, and it looks like it works. And then next thing you know, the cancer's relapsed, and now it's resistant. And very much of the literature says, well, cancer's very heterogeneous, and you just select for resistance. But I had sort of wondered whether if you could translesion DNA polymerases be copying over the lesions and making them into mutations. And when Mike Heeman came, tried this idea out on him, and Mike, my great joy, was willing to give it a shot. And that's just opened up a new area. Not only did it prevent, in the lymphoma model, if we knock down the pathway, did it prevent the relapse, tumors getting resistant, but we took Tyler Jack's KRAS P53 minus mouse model of non-small cell lung cancer, and this is famously resistant to cisplatin, but it turned out if you took out one of the components of the mutagenic translesion synthesis, I think you can see just from here the tumors did well. So we've had really nice structural collaborations lately. The business part of Rev1, that it's got a recruitment domain that's 100 amino acid 4-helix bundle at its extreme C terminus that recruits the Rev7 component of the Rev3, Rev7 polymerase, which is called Pol Zeta. And there's a key little interface right in the middle of that circle. And I'm pretty excited right now because in collaboration with Peju at Duke and looking at, there's another interface as well with Dmitry Korzunov in Connecticut. We've been looking to see whether we can get small molecules and inhibit them. And Peju's lab and we and Nimrat Chatterjee in particular we've got a small molecule that seems to block that interface, and it's taking down the pathway, and we're getting a lot of really interesting results. So we'll see. But anyway, it's a direct present-day consequence of this earlier work. The other thing on the DNA repair side, Cynthia, her second damage-inducible gene that she found with her like fusion, called it DIN-B, turned out to be DNA polymerase 4, a member of the Y family. And Dan Yarish, who's now a faculty member at Stanford, and Veronica Godoy, who's a faculty member at Northeastern, when they were in the lab, found out that this polymerase can copy preferentially over into guanine adducts of a certain size quite accurately. So it's a different kind of way of polymerase acting. And it does it way better than over an ordinary guanine, as if it was specially put in there. So we were looking at that. And Jamie Foti in particular got wondering, why was it when we overproduced the polymerase, it was lethal? And instead of just taking over, because it's a very slow polymerase. And I can't go through the science, but it ended up leading us to the discovery that what was happening in those cells, there was oxidative stress, 8-oxo-DGTP was getting oxidized to 8-oxo-DGTP. It was getting incorporated. You were getting partial base excision repair, and these base excision repair intermediates were leading to a form of DNA damage. And this whole thing reminded me of the work that Jim Collins, who was at that point at Boston University, had been doing on antibiotics. And we found that that same mechanism applied. And it's a great joy for me that Jim has been here and our collaboration has continued. There's a great deal of evidence supporting this now. And most recently, Charlie Gruber, who's sitting right there, we were able to show that other forms of stress cause lethality by the same mechanism. And in that case, it was one of these fusions John Beckwith used years ago, fusion of the periplasmic MALE to the cytoplasmic LAC-Z, which makes the cells die, but it turns out it makes them die not by jamming the membrane, but by causing this whole sequence of events that leads to damage in DNA. So that's kind of how the DNA damage thread went. You might wonder how I ended up working on Rhizobium as well, but while I was a postdoc, two significant developments happened. One was transposons became a daily tool, suddenly a tool in bacterial genetics, and I realized even organic chemists could do genetics if you had the transposon to work with. And recombinant DNA was developed, so I thought, wouldn't it be fun to take a bacterium that is something other than E. coli and see what we could do with that. So this is one of the places where my teaching and my research have intersected, and there have been many of them in my career. But I had been teaching project labs using this mu phage to make LAC-Z fusions, and I thought, let's do a Rhizobium lab. But I wanted to have some projects in my back pocket, and I talked to Mark Van Montague, who worked in agrobacterium, about a bunch of just ideas, and I learned that agrobacterium fluoresced under UV on plates that had chalcoflora, which is a laundry whitener. And I thought, oh, undergrads would like glowing colonies. Let's see if our strain, Rhizobium strain, fluoresces. It did. The idea is that when the chalcoflora binds to a beta-linked glucan, it absorbs UV, emits invisible. And so I persuaded John Lay to make some mutants. John's been at the University of Washington for many years. And it turned out all the mutants that didn't fluoresce were broken in symbiosis. And what actually happened was they had trouble getting down the early stage from a curled root hair down and coming into the developing nodule. I guess I skipped one thing. I picked Rhizobium because I thought this was a good bacterium to do. It was kind of like E. coli, but it did all this really interesting stuff with the plant. And I wasn't so interested in the nitrogen fixation part, but I really was fascinated about how it managed to get in from the outside. And then in the inside, it eventually penetrates into the developing nodule. They come down this little tube of plant origin, and they basically get endocytosed into the cytoplasm. And I thought, well, those were two really interesting and poorly understood aspects of the science. So let's see here, okay. Anyway, it turned out the molecule that was needed to come down the infection thread was a complex polysaccharide called succinoglycan. And we worked on that for a long time. Another surprise came, we found a mutant that called BAC-A that blocked the cells once they had been cytosed. And I wondered if any of these functions might be in common with chronic intracellular mammalian pathogens. And Brucella was the one we looked for, and we found, in fact, that was true. And my former grad student, former postdoc, Gail Ferguson, went on to show that BAC-A gene plays a role in symbiosis. It seems to affect the ability of the cells to withstand sort of defensin-like peptides that are made by the plant to force the bacteria into a terminal differentiation once it gets inside the cell. And Gail, unfortunately, died of metastatic breast cancer just a small number of years ago, and I want to make this tribute to her. She was an amazing scientist and a huge part of my group. And then the other fun thing about working with Rhizobium, and Tom sort of alluded to this, was maybe it's just using another model system, but there have been some unexpected findings. This same screen, which we use to look for non-fluorescing calcifluor, mutants on calcifluor plate, we also noticed bright ones, and some of these bright ones were pretty profoundly symbiotically defective. And thanks to the work of first Gordon Campbell and then Michi Taga in particular, we ended up discovering that the function of that gene was, of all things, necessary to make dimethylbenzimidazole, which is the lower ligand of vitamin B12. And that was significant because despite four Nobel Prizes for vitamin B12, there was this gap in the synthesis. Didn't know how to make dimethylbenzimidazole. And so we collaborated with Chris Walsh and found that this one enzyme did this magic little trick of cutting 3 bonds and forming a new one to make it out of reduced FMN. So that was one sort of surprise that I thought it was working on symbiosis, but the great joy of science is sometimes working on something else. And Tom alluded to this one. Brian Davies, who was an intensely creative grad student with me, had an idea for a screen and got some new symbiotic mutants. And one of them caught our attention because it was in virtually every bacterium in the world. It was called the putative metal hydrolase. And we started to work on it. And this is partly where Tom and I, after having been friends and colleagues for many years, really got to collaborate. And we now know that this gene, which we call YBY, plays a critical step in the 3' processing of tRNA, in 70S quality control, and small RNA regulation. Again, one of the great joys of science is you just never know what you're going to find. So I just say briefly, I love teaching. This was me recreating something from my introductory biology lecture for the American Cancer Society donors at some event, I guess. But I was heavily influenced by the project lab teaching that we did, and I was sort of trying to, I think, light a candle in people, see if you could get people excited about doing research. I was asked to stop teaching project lab when introductory biology became a required subject, and I've been teaching since 1995 with Penny Chisholm. She's the one on the left. That was when she got the Medal of Science, and we tried to do a 701 7014 that has a little bit of an ecology and evolution spin to it. And then when Salvador Luria retired, I took over and taught with microbial physiology with Boris for many years, and then Tony took over recently, and Kevin was just our TA for this past semester. As Tom alluded to, I was undergrad officer for, I guess, a total of 22 years, and that included some time when we had, like, at one time we had 186 majors. I have a memento that I have to share with you. When we moved from building 56 to this building, I got a piece of my lab bench that says that this piece of lab bench is rescued from the Walker Labs in building 56 on May 15th, 1995, during the gutting of the old biology building. It was rescued by a stout-hearted crew of undergrads in appreciation for what you've done. And so I had some wonderful times with that. Somewhere in '86, just before our son was born, we were asked to be houstmasters at McCormick Dormitory, and whether it was a good idea or not, we did it, and we did it for 6 years. Our son grew up in a dormitory with 250 women. He had 250 big sisters, and I learned to use the pizza skills, expand them up, and make 35 cups of flour into pizza dough, and have 75 people over, or how to do barbecue, a very limited budget, how to do a barbecue for 200 or 350 people, where I had one of my best ideas at MIT ever, which is to rent a cotton candy machine, which you should do sometime. And for many years, I directed, for 25 years, I indicated we'd had this wonderful grant from Howard Hughes that supported the development of Europe, the development of the MSRP program, many innovations in Project Lab, and some high school outreach. And that was a huge thing in getting all of those programs established. But then I was asked to compete for a Howard Hughes professor, which was something individual, and I didn't end up wanting to compete with myself. And I want to acknowledge that Mundana was my co-conspirator for most of those years on the Howard Hughes thing. So I didn't want to compete with myself, so I had the idea of starting an education group that was sort of modeled after a research group. And I had it once for 4 years, and then a little gap, but we continued with some other money, and then another 4-year period of funding. And although we did a lot of things, the part I didn't anticipate would happen was we'd start interacting with some of the programmers at MIT and would produce 3 forms of biology education software that's been widely used in other universities around the world, in high schools, and even in Haiti, where all the educational infrastructure collapsed. And Mary Ellen is sitting here, who's been very, very active in all of that, and even helps me when I use some of these in my 7014 class. Just almost done here. Just a couple more things I thought about. I did take a sabbatical every time. I was inspired by the way Maury Fox had done that. Maury had introduced me to Dick Dari in Paris years ago, who became to be a good friend, and I had a lot of scientific meetings in France at the time, and so I had a sabbatical with Sharon Long, where I worked like a grad student in the mid-'80s, and then one later in Geneva, Bill Broughton, who was a Rhizobium one, but the one that was so influential was one we had in Paris for 7 months in 1994, when my son was about 7.5, and I thought it was a really bad idea as we were leaving. It just seemed overwhelming what we were about to do, but it ended up being a wonderful experience. And doors opened, and as Tom indicated, I was editor-in-chief of Journal Bacteriology for a lot of years, but then Errol Friedberg twisted my arm, I don't know exactly how, to co-author this book on DNA repair and mutagenesis, and then the first edition, and then a second edition. And for those of you who don't know this, it was about 5 years worth of work on each one, and most of the time it looked like that. There was always piles of papers trying to get my head around and absorb all this literature and figure out how to make it into a textbook. But it it was really a lot of fun. The other thing, I guess, that door opened was Phil Hannawalt, who I mentioned, had started a series of community-wide DNA repair and mutagenesis meetings that I thought they were the best science. They brought everybody together, and they were so much fun. So I eventually sort of picked up that torch, did the first one in Hilton Head in 1999 with Errol Friedberg and Susan Wallace. It's Priscilla Cooper who helped me organize the next two. The next one was in Bermuda, and the final one was in Whistler, in which Leona Sampson almost helped. I think about 1,500 people came, and I tried to do some of the things I wanted to do, apart from really great science and everything. We had a lot of dancing, but we also had music, and I had a friend, Rick Wood, who's very noted DNA repair biochemist, who's also a virtually professional bass player, who helped us do an open mic. So we had homemade music at all the meetings, and I felt a lot of my career came together in the Bermuda meeting, where I decided to, at the end of the opening session, I wrote a song called DNA Repair Blues, and Rick showed up with his bass, and Errol Friedberg, the author, plays didgeridoo, who knew? And I got Phil Hannawalt, Susan Wallace, and Sulla to help sing, and Mary Ellen was there. I think it was a great time in my life. We just had our 40th lab anniversary. I can't thank all the people who worked in my lab enough. It's all I think I do is sort of try to help now tell the world about all the creative stuff that they have done over the years. But it's been a joy, and I think it's by and large been like a family with everybody supporting each other. And I couldn't have done any of this without my wife Jan and then my son Gordon, and so I feel grateful to them, and I'm just so lucky that whatever that star was that hit me on the head, I got to be here and eventually got to tell all of you about it. So thank you. You don't have to ask questions, but if anybody's got any, I'll be more than happy to talk.
So a bit more yours.
Well, I'm trying to figure out that part of my career as well, so we'll see. But it was a great honor to have gotten that award. Yeah, and will help with chasing down some of this most exciting of the DNA repair stuff and the chemotherapy angle, I think, particularly, whether that might do something. Yeah.
Somewhat of a philosophical question. You've certainly recruited some amazingly creative people, and over the years done amazingly creative work, and these people have gone off to do wonderful things. What is it that you feel in your environment, or what is it that you promote in your environment that keeps this cycle going? I mean, it's really challenging to recruit and keep good people, and especially in science these days. What do you think it is about your mentoring style, or?
Well, yeah. I mean, I've not really been so good at recruiting people. I've actually, because I've been working on systems that aren't very hot, often someone will show up and I'll say, great, you want to work with me? So I haven't done a lot of discriminating compared to some of my colleagues. But to answer your question, what I say to people, and the way I truly think about it is, I said, I think the most important thing I do is believe in people until they can take over and believe in themselves. I think virtually everyone I've seen has in them an intrinsic ability to do really creative stuff. And my experience at the grad level, postdoc level, and at the undergrad, which certainly informed me as well, is usually when people have troubles, it's not that they're smart and they're not creative. It's just life is very complicated. And all kinds of other stuff gets tangled into this. So your confidence gets shot, or you can't focus because of this problem or another. So I think for most of the people who've done well, or for many of the people I've done well, I know I made a contribution, but it isn't what anyone would think. We just kind of quietly helped sort out some part of some complexity in their life so they could get free to work again. And then they flew. And I try to, my best to give people as much independence as they can use and want, and maybe a little bit more at that point, but to try and mix it with being there to help out with problems, because I guess my goal is when somebody leaves the lab, I want them to have strong wings and feel like they can fly without me being there, or with just me being there, continuing to cheer them on. I guess maybe that's kind of the way I think about it. Okay, well, thank you all for coming. It was a real pleasure. Thank you.